Okay. Good evening, everybody. Good evening, everyone. Thank you for being here, of course, today. My name is Keith Mwanzia, and I'm the president and CEO of the Guelph Chamber of Commerce. And I get the chance to welcome you today, and of course we're quite happy to welcome you here because, of course, Guelph is the birthplace of the DNA barcode. And I was speaking to someone and said, "How many times have you heard that this weekend, or this week, I should say?" And apparently, it's not very many times. So I believe that there was a sign on the way in that should have said, "At least 5 times you have to say that this is the birthplace of the DNA barcode." So count it on one hand or both how many times you've said it. I had the chance to interact with a number of you during the reception, and the reception that we hosted alongside with the city of Guelph was what we called a taste of Guelph. It was an opportunity for you to savor a little bit of what we have to offer when it comes to music and food. But as you looked out of the River Run Center and saw how beautiful a facility this place is, or the scenery around it, we're reminded about, of course, how important the work is that you do, and how important continuing to tackle this particular issue is for communities like ours and is for communities around the world. I was also very happy to see that not only are we hosting you in the birthplace of the DNA barcode, but we're also extremely happy to welcome people from 68 different countries to our community. And I think that's worthy of applause. Over 500 delegates from 68 different countries. That's fantastic for our community. We're so pleased to welcome you to our community. We're so pleased to welcome you to Guelph. But what is more significant is that this work is being tackled in 68 different countries around the world, and that you are converging here to connect, and that you are converging here to be able to engage about how there can be a common thread between what it is that you do and what applications we can see for it in the world. I'm also particularly excited to see that there are people from countries large and small. There are people from countries of various economic diversification. And that says to me that this is an issue that is not only topical to individuals and communities and businesses, but this is a world issue that all of you are tackling in a meaningful way. So I thank you for that. I thank you for coming to our community, and I encourage you. I encourage you to check out more sights and sounds of Guelph, explore our downtown, explore our trails, participate in a community that truly has had the chance to welcome you in a meaningful way. Last, but of course not least, I would be remiss if I didn't take the opportunity to ask you to join me in thanking the University of Guelph for their leadership. What they have done is incredibly meaningful, not only in this community, but their involvement in convening this type of conference, their involvement in elevating our community, both business and alike, to an international stage in a meaningful way, is central to how the university contributes to Canada, to Ontario, and of course to the community of Guelph. So please join me in thanking the university. So it is without further ado that I will introduce someone that might be able to offer more of an academic, a conversation to you. But it's been a pleasure engaging with you in the reception. I look forward to the talk later today, and it's truly fantastic to have you here. I welcome you many times over, and once again, I'm excited to welcome you to the birthplace of the DNA barcode. Please welcome Carl Tibelius, Vice President, Genomics Programs at Genomics Canada.
Thank you. It's a pleasure to join you all today. Such a great pleasure to be here in Guelph, one of Canada's most welcoming and beautiful cities. Luckily, I have lots of reasons to come here. Both my daughter was educated here and still lives here, and of course my visits to the university to visit Paul Hébert and others. So Guelph is a community where nature and the outdoors are never far away, and Guelph is home to a great university that develops groundbreaking knowledge on the nature of life. So I want to begin by thanking everyone who lives here in Guelph for hosting this exciting conference. The true value of genome science lies in helping people live better and healthier lives, and in protecting the ecosystems that sustain all life. And the 6th International Barcode of Life Conference is a valuable forum to share and promote such knowledge. Since 2003, DNA barcoding has become the largest research program in biodiversity science. What is encouraging about this conference is that with each iteration, it grows in scope and ambition. This year's theme, Barcodes to Biomes, continues this tradition by broadening the study of species in particular places to understanding biodiversity on a regional and a global scale. Genome Canada is pleased to be a partner in this endeavor. In fact, we are proud to be lead sponsor for this special plenary session this evening on the state of biodiversity. Genome Canada has been a big supporter of DNA barcoding for nearly 10 years. From 2006 to 2009, we supported the Canadian Barcode of Life Network, investing more than $5 million. Genome Canada and the Ontario Genomics Institute then worked in close collaboration with Paul Hébert and his colleagues to develop a larger, more ambitious international barcoding project. And beginning in 2009, we have funded the International Barcode of Life project, investing more than $16 million into this initiative since that time. The Ontario government, the University of Guelph, the Canada Foundation for Innovation, and the International Development Research Centre, amongst others, have been valuable partners. International partners included institutions in Germany, Argentina, Mexico, China, the USA, and many, many other countries. The result is an achievement that all Canadians and all of humanity can be proud of. The largest biodiversity genomics initiative ever undertaken. Thank you, Paul, for your vision, leadership, and the very hard work of your entire team. Today, collaborators from over 25 countries take part. The Barcode of Life database here in Guelph now contains more than 5.8 million specimen records representing over 500,000 species. You set bold objectives for 2015, and you achieved them. As many of you know, this is fascinating science that serves a very direct purpose. The DNA Barcode Reference Library has very practical applications. It helps ensure consumers get the food products they paid for. It helps stop the trafficking in endangered species. It helps fight invasive species and protect ecosystems, natural resources, and agriculture, and is being used to make mining and other activities more sustainable. These benefits align with Genome Canada's mandate and mission. We are a not-for-profit organization that acts as a catalyst for developing and applying genomics and genomic-based technologies in order to create economic and social benefits for Canadians. We connect ideas and people across public and private sectors to find new uses for genomics, invest in large-scale science and technology to fuel innovation, and translate discoveries into applications and solutions across key sectors of national importance, including health, agriculture, forestry, fisheries and aquaculture, energy mining, and the environment. This conference is a great opportunity to explore other broader applications and to engage the wider public. So to those of you who have joined from that wider public, I hope you will meet some of the scientists and ask questions and continue following progress in genomic science. Thank you, everyone, and I'm sure that you will enjoy this evening's presentations by some of the world's most influential biodiversity scientists. And just before I leave the lectern, I have the pleasure of introducing the chair for this evening's section, Brock Fenton, who's Professor Emeritus at the University of Western Ontario. Thank you, and over to you, Brock.
Thank you very much. Good evening. We're going to give you an experience of hearing different people's views about biodiversity, what we can do about it, and perhaps various intersections it makes with barcode analysis. And this is something that a lot of us have been listening to this week. I'm going to begin by just giving you an example of how barcoding has changed the way that bat biologists look at the world, or at least some bat biologists. You appreciate that bats are really important. There's about 1,200 species in the world today, and there's many different things that these animals are thought to do or actually do. The first fossil bats appear about 52.5 billion years ago. They're already perfectly good bats. By 52.5 billion years ago, there were already 10 families of bats. And one of the interesting questions, of course, would be, how did they get started in the first place? And of course, where and when did that happen? I doubt if barcode analysis is going to tell us much about that. We do know that there's rich interactions between bats and the insects that they eat, and this often leads to some interesting nocturnal warfare, with bats locating insects using echolocation or biosonar, and insects using those sounds against the bats to minimize their chance of being caught. The trouble is that if you look at a bat turd, such as the one on the right, you're looking at a horrible jumble of pieces of insects. And it was Beth on the left, not the hairy one, that in fact had the brilliant idea of picking pieces of insect out of bat crap and doing barcode analysis, so that instead of bat biologists saying that bats eat insects, they eat moths and beetles, we can actually prepare a list of what they eat. When we do that for two common species like little brown bats and big brown bats, which are different by a factor of 2 with respect to body size, perhaps not surprisingly, we find some overlap in terms of what they eat. So the idea bat biologists have had for some time that bats compete with each other, and it's competition that drives speciation in bats, DNA barcoding has pretty effectively derailed that point of view. The interesting point, of course, is that bats are very high energy animals. They consume huge amounts of energy. So we have looked at a community of insectivorous bats in Jamaica, and we find that there's relatively little overlap in terms of what the bats actually eat. There's no evidence of competition, but they eat a vast number of different kinds of insects. One reason for this is that if you look at the situation facing a female little brown bat, she's eating 75% of her body weight in insects every night, and she has to produce copious amounts of milk because her baby is eating its own weight in milk every day. To put that in perspective, a 45-kilogram woman lactating would be producing 33 kilograms of milk a day to feed the offspring. This puts the bats in the position of eating more than their own body weight in food every day. To scale this, my friend Eleanor here is giving a demonstration. She is surrounded by her weight in pizzas. And as much as she liked pizzas, that was a little bit more than flesh and blood could bear. So it's perhaps no wonder that there's overlap in what they eat, and perhaps what bats are in is just a business of who can get the most food the fastest, and there seem to be lots of insects around. Flight and echolocation were two key factors in the diversification of bats, and to date we don't have any evidence of competition, and DNA barcode analysis has given us powerful new tools for looking at the details of what bats actually do. We discover, for example, that bats such as these from the biodome in Montreal are said to be nectar-feeding bats, but in fact, when you look at what they eat, they're actually eating a lot of insects and they're eating a lot of fruit. They are actually very well connected in the ecosystems where they occur. So all of a sudden our change in our view of these animals changes as well. And of course, for bats that live in the tropics, this relates directly to their role as pollinators of plants and to their role as dispersers of fruit. So it turns out that the barcode analysis has changed our view of these bats, and I suspect that that's just going to keep on happening, and we've heard about that sort of thing repeatedly over the last couple of days. So the whole point of the it being a sea change to what bat biologists do. It's not that long ago when somebody said, what does that bat eat? Referring to a big brown bat. You said insects, perhaps mainly beetles, because there was little prospect of being able to identify what they actually ate. So barcode analysis has effectively turned the bat world, I think, on its ear. Speaking of ears, bats' faces are often reflective of the challenges of biosonar, which doesn't have very much to do with barcoding. So that was my way of getting us started, looking at the way something like DNA barcode, it can revolutionize the way you do your science. We're next going to hear from Lawrence Packer, who's going to talk to us about bees, and he's waiting in the wings.
Hello. I'm no longer waiting in the wings. I'm Lawrence Packer, and I like bees. I like bees a lot. And I don't have much time to tell you about them, but I'll say something about why they're important. There are many other reasons bees are important than the ones I have time to talk about, but I'll talk about a couple. Then I'll talk about the use of DNA barcoding to try and discover new species of bees. There are 20,077 known species of bees on the catalog. I've got several hundred in my office waiting for me to have time to describe them. So the total number is probably close to 25,000, and DNA barcoding is certainly helping a lot of us discover that kind of diversity. And then I'll talk about how far we've got in terms of the campaign to get DNA barcodes for all the bees in the world. I'm a kind of ambitious young man. Okay, so we all know that bees are in trouble. We hear something about this every week. There's pesticides, which are a problem. There's habitat fragmentation. There's climate change. I'm not so sure cell towers actually are a problem for bees, but bees are in the news a lot. Bees are in trouble for a lot of reasons. One of the reasons they're in trouble is most people think they know what bees are. This is a book called The Bees of the World. That is a fly on the cover. The people that wrote this book know very well that this is a fly. They are rather upset with the publishers and they complain, and the publisher says, no, we've checked with entomologists and they tell us it is a bee. It is a bee. Okay, so the main thing we think about when we think about bees, I think, is the fact that they're important for our food. So if the bee disappeared off the surface of the globe, man would have only 4 years of life left. Woman, being more adaptable, might last for 6. Einstein scholars can find absolutely no evidence that he said anything of the sort, but what we can see from this is that he had absolutely no idea what bees look like. So here's breakfast with bees. That looks kind of nutritious. I should have put some milk in there. 15% of milk and dairy, even if you don't eat any fruits or vegetables and you only eat meat. 15% of meat and dairy productivity in Canada is brought to you by bees through pollinating alfalfa, which is winter forage for cattle in many parts of the country. So here's breakfast with bees. Here's breakfast without bees. Doesn't look quite so interesting. We still get coffee. Coffee self-pollinates, but the yield is so much higher when there are bees around to cross-pollinate it that there'd be far more people grumpy in the morning if bees weren't around, because it would be so expensive that many more people wouldn't be able to afford it or afford so much of it. Okay, so when people think of bees, they normally think of the honeybee. That's the one we put all our eggs in that basket. Where the data are really good, which is England, only 30% of the agricultural productivity that requires pollination is pollinated by the honeybee. It's wild bees and to a lesser extent flies that are doing the rest of it. To get a North American example, watermelon. To get a nice healthy watermelon, 2,500 pollen grains have to be transported by a pollinator from the anthers of one flower to the stigma of another. In a study in California in an organic watermelon field, there were 42 different species of bees that were doing that, and they did an excellent job of it. Rather worryingly, as the habitat deteriorates, it's the better pollinators that are disappearing first. Another great story associated with this is that it's not just the total number of visits that's important, it's getting visited by multiple species. In other words, 10 visits by one species transfers a lot less pollen in a way that's useful for the watermelon than one visit by 10 different species, each of 10 different species. So bee diversity is really important for agricultural productivity, even though most people only think about honeybees. Yes, in terms of conservation, honeybees and all other bees are impacted by all the kind of things that we hear about: pesticides, climate change, habitat fragmentation, and the rest. But first, what we've got to do is to find out actually what they are. So now I'm going to give you a couple of examples of using DNA barcoding to help us identify bees to the species level. I did my PhD on this species. The title was Geographic Variation in the Social Behavior of. The title's now known to be wrong, because it wasn't geographic variation, it was variation between two different species. It used to be one of the most easily identified bees in North America. Its head is really triangular in the side because it's got kind of pointy chin, and it's got these little white bands on the abdomen. Lots of bees have got that, but that combination is unusual. It's also got very large mandibles. Its bite is as bad as its sting. So, we now know that it's at least two, well, we soon found out after I finished my thesis, that there's actually two genetically distinct different forms. One is found in the southeast, as we see here, Florida, and along the coast towards southeast Texas, and up as far north as close to Washington. And you find the two species together in the southeastern foothills of the Appalachian Mountains. They are morphologically identical. Nobody's been able to find anything that separates them using standard morphological methods, even morphometric analyses of the male genitalia, which, if you're an entomologist, you'll know how much time we spend observing those parts of male bees, because it's usually very good at separating closely related species. Even taking minute measurements of the naughty bits doesn't help us separate these two species. Nuclear genes, mitochondrial genes, and proteins separate them extremely well. There's almost 6%, the light battery must be running out on this, there's a 6% or so sequence divergence between the one species on the top and the one species you see lower down. And then it occurs through Mexico into Central America, and recently we found that those actually indicate even a third species. So what used to be considered the most widespread bee almost in the world, easily identified, now becomes three different species, not so widely distributed and impossible to identify without genetic information. Second example is, this takes me to one of my favorite parts of the world. The weather is usually good in the Atacama Desert. It doesn't rain much. And it has some really rather beautiful bees, including species in the genus Loncopria. 3 species were known in this genus, but with the DNA barcode suggests to us that there are 6. So, okay, in the previous example, we had nuclear genes as well as proteins that added evidence to support the barcode differences. What have we got here? Well, the way morphologists tell these species apart, this was discovered by Haroldo Toro, shown here, is that unusual for most bees, during mating, the male holds onto the female, the base of the female abdomen with his mandibles, and they fly around like that for quite a long time. As a result of that, there's been strong sexual selection for the shape of the male mandible and the shape of the female receptive parts at the base of the abdomen. And so we can see that pretty much uniquely among the bees, different species have got completely different shaped mandibles, and the females have got these strange, quite unique structures on the side at the base of the abdomen that show that the male mandibles fit into. So in the paper, they describe this, and I'm not quite sure what's going on here, but it is the mandible fitting into the female part. To me, this reminds me of one of the most famous paintings of the last century, Marcel Duchamp's nude descending a staircase. So we've got a species that looks like another one. We know what parts to look at in this case. We look at the mandibles. And here is the one that was described, the drawings on the right, and here's actually a photograph of it. And the one that's undescribed has got a completely different shape to the mandible. And once I get the time to look at the female, I'm pretty sure I'll be able to find that the sockets that that mandible fits in are also different in shape. Now we get to an example where the number of species is going to go up enormously. These are really cute little bees with remarkable sexual dimorphism. That's a male. You wouldn't guess that that's the same species as that one just by looking at it. The color and shape's completely different. The male's got this flat head and very bright color pattern, and he sits on a stone looking up for anything that might be a female flying overhead and then flies towards it. There are 29 species of these known from Chile, described by Luisa Ruz, shown in the top there. We have barcodes that indicate we've got at least 32, probably more, different species level barcodes, but only 11 of them key out to identified species, and all these red numbers here indicate a distinct barcode that's a different species. And Luisa, the world expert on these bees, she's looked at them and she can't find any difference. She can find differences, morphological differences. She can't identify the barcoded ones that come out as not currently known species. So in this genus, we've probably got twice as the number of species as there are already known. Okay, to take a completely global perspective now, in 2008 at York University, we held the first meeting, actually an only meeting, of the people involved in campaigning to barcode the bees of the world. If you've ever organized a symposium, even a small one, you might notice everybody in this picture there is smiling except me. Organizers, you have my sympathy. I know what it's like for a small gathering. For something as big as this, oh my goodness. So, as of 2013, these are where we had barcodes from. We've been remarkably successful in getting these from pretty much all over the world. We've even got a barcode from a specimen somewhere where there are no bees. There aren't any bees in the Antarctic Ocean. Somebody put the latitude and the longitude the wrong way round. But this leads me to suggest where I would like you to go on holiday to collect me bees. Okay, Siberia, Saudi Arabia, the Sahara, the middle of the Amazon, in the middle of Australia also. So we've done really quite well in getting global coverage. And this is 2 years old now. It just takes too long to do it for 2015 because there's so many records in the system. So what is the geographic coverage in comparison to the number of species we know occur in a particular place? Well, if we look at Thailand, for example, there's less than 200 species known, but we've got barcodes for over 220 of them. And that's because a friend of mine was running Malaise traps in national parks in Thailand and sampling the biodiversity of that country very effectively. In Canada, where we've done the most intensive collecting, we've still only got 3/4 of the number of species. That's because there's a large number of species only known from the original specimen, things that are extremely rare. We're suffering from the law of diminishing returns. In Ghana, just a couple of field trips and a few collections by locals, and we've barcoded as many species as are known from the country. In Oman, one of my graduate students went on a 2-week field trip, and he got almost all the bees that are thought to have occurred there. Obviously, the diversity of bees in Oman is much lower than it's expected. Britain. Look at that. You know, a society in disarray. You know, they've got 260 species of bees, and they've barcoded maybe 3 of them. Okay, so overall, so far, we've got DNA barcodes for over 90% of the 500 different genera of bees. We've got about 30%, the number of distinct barcode clusters we've got of the 20,077 species is about 30%, which is remarkably good progress in a short period of time, less than 7 years, really. But we still need help. And this is why I'm encouraging you to go on vacation to Mali and the Yemen and some of these other lovely destinations for calm, relaxing vacations. All you need to do is to get some brightly colored plastic bowls, put soapy water in them, put them out in the ground, go for a walk, put another one, put another one, put another one. And then at the end of the day, you walk back and you strain the bees out with a tea strainer, put them into ethanol. Oh, it's difficult getting ethanol in some of the countries where bee diversity is high. Rubbing alcohol will do. They will let you have that. I don't suggest you drink it. And then send them to me, and I'll be forever grateful. And one of the things is I've got so many species to describe, and from these places you will find many more. I can even name one after you. So the work that we've been doing has been funded, as it should be, by a very large number of different organizations shown here. A large number of people have helped us. The number of collaborators that have sent us bee specimens now is just about every bee researcher in the world, with only one or two exceptions. So we've got a lot to be grateful for in terms of the funding and community support. And lastly, I'll thank you for listening. How am I for timing?
Perfect. We're going to give people a chance to ask 2 questions.
Yeah.
We're going to have time to take 2 questions of speaker before we move on to the next speaker. We have 2 people with microphones, so if you have a question, put up your hand and someone will put a microphone in it.
I know Barcode has not been designed for much of phylogenetic, but did you get any phylogenetically sensible trees out of your bees? Did phylogenetic groups stay there as clades?
Okay, good question. Do we get sensible clades and phylogenies with DNA barcode sequences? I remember once I analyzed all the data I had from the bees of Mexico, and the results fit the phylogeny perfectly. And then I started analyzing it using rigorous phylogenetic methods rather than neighbor joining trees. And a lot of that pattern disappeared. When I added in taxa from elsewhere, a lot of the pattern disappeared. But my friend Christoph, where are you? Christoph here has got a lovely data set that shows you only need a tiny amount of nuclear DNA, add it to the DNA barcode, and you get results that are sensible. You agree with what I just said, Christoph? I didn't paraphrase it too badly. So if we can get a nuclear barcode as easy as we can as the mitochondrial barcode, we'll be able to have phylogenies for 30% of the bees in no time.
Another question. You're letting them off too easily. Thank you very much, Lawrence.
I normally get rough questions.
Thank you very much. Fantastic. Our next speaker has chosen to eschew the modern things of PowerPoint and whatnot, but Monty Hummel, who's about to arrive, he's just arrived. Monty Hummel's going to talk about conservation.
Thank you, Brock. Good evening, everybody. The underlying theme of my talk is that if we want to conserve species, we need to conserve that which makes species possible. As a lifelong activist, I'm less interested in documenting the current species loss situation than I am in doing something about it. But make no mistake, globally speaking, we are losing. In WWF's most recent Living Planet report, based on expert analysis of more than 10,000 representative populations of mammals, birds, reptiles, amphibians, and fish, we estimated there has been a 52% decline in all species populations worldwide since 1970. Terrestrial species declined 39%, marine species also 39%, and freshwater species a whopping 76%. For over 40 years now, humanity's collective demand on nature has exceeded what our planet can replenish. As a result, we would need 1.5 Earths to meet our current demands. In other words, Humanity is now eating into our natural capital, thereby jeopardizing the well-being of both current and future generations. The practical challenge for us all is to more equitably improve human well-being while keeping our ecological footprint within globally sustainable levels. To do this, we need to protect natural capital, what this conference is about. We need to produce better, we need to consume more wisely, and we need to redirect financial flows and investment. And I would add, we need to voluntarily control human population growth. Human well-being ultimately depends on biodiversity and natural resources such as wood, water, arable land, and fish, and on ecosystem services such as pollination, nutrient cycling, and erosion control. Over the last 40 to 50 years, conservationists have deployed 3 general strategies to conserve these ecological services and the species they encompass. First, protect. Protect species and ecosystems that are still in a healthy wild state so they can fluctuate and change as much as possible under natural conditions, hence nature reserves and protected areas. Second, manage. Manage the human use. Notice, not manage wildlife, manage the human use of those species and ecosystems that are going to be exploited so that such use is sustainable and based on sound conservation principles. Hence, such international initiatives as both the Forest and Marine Stewardship Councils. Third, restore. Restore those species and ecosystems whose backs are against the wall through degradation, overexploitation, or the threat of extinction. Hence, WWF's efforts to help recover the Grand Banks off the east coast of this country where the northern cod was so tragically overexploited. Now, traditionally, we conservationists have found it easier to emphasize protection and restoration over conservation management. We are instinctively preservationists more than we are conservationists. But my first point tonight is that we must become as enthusiastic about announcing, for example, that 100,000 hectares of forest are going to be managed sustainably to the international standards of the Forest Stewardship Council, as we are enthusiastic to announce that 100,000 hectares are going to be strictly protected in a park. Because both announcements are necessary, and both make a contribution to biodiversity conservation. Maybe not equal contributions, but important contributions nevertheless. Therefore, we must stop regarding lands that are going to be logged, mined, and yes, even drilled, or waters that are going to be dammed, used for irrigation, or commercially fished as ecological write-offs. They don't have to be, and they no longer can be treated as simply trashed. That's why WWF's current conservation program in Canada focuses on both wild and working places. Now, I don't know, maybe all this seems rather obvious to this audience, but it's quite controversial in conservation circles and is proving to be a somewhat challenging pill for more protection-oriented activists to swallow. It's just easier to be more precautionary and to always insist on strict protection than to allow the more complex boogeyman of sustainable use to get his foot in the door. To this, I say, too bad. Suck it up and engage the real world. My second and related point is that we must go deeper, which reiterates my theme of conserving what makes species possible. We all know that if you want to conserve species, you have to conserve the spaces or habitats upon which they depend. And then go deeper to conserve the ecological processes and maintain the ecological processes that make spaces and habitats tick. But to really understand this, it may help to think of species as essentially ephemeral. They come and go and change over the fullness of time. The flora and fauna we now witness on Earth are in fact a product of the interaction between climate and certain fundamental physiographic and biochemical elements of our lands and waters. Things like landforms and underwater topography, soils, currents, geology, parent material, and nutrient potential. It follows that if we want to conserve a representative sample of the species on Earth at any given time, we must identify and protect a representative sample of that which makes them possible, what conservation biologists call the enduring features of our lands and waters. So to cite a fun example, let's say climate change means that alligators someday occupy the Hudson Bay lowlands in the subarctic region of this country. Well, if we've protected representative and intact samples of the enduring features of the Hudson Bay lowlands, we will also conserve a representative sample of those alligators, plus whatever other animal and plant species that might have evolved on that particular landscape under a changed climate regime. Now, needless to say, this approach is also controversial in conservation circles, not so much because the science isn't sound, but because I think some activists feel it is too adaptive, too fatalistic, almost like giving up and accepting as inevitable human impacts that we must try to head off or change. But actually, what I'm sketching is a potential pathway to resilience. It's an intelligent basis upon which to locate protected areas and other species conservation initiatives, regardless of what happens to climate. And in no way am I suggesting that we sit back and do nothing about the outrageous impact humans are having on climate and therefore on our planet writ large. Because carbon from fossil fuels has been the dominant component of humanity's ecological footprint for over 50 years, and it continues on an upward trend. Again, in the Living Planet report, we determined in 1961, carbon was 36% of our overall footprint, but by 2010, it comprised 53%. That is precisely the kind of systemic impact that threatens to undermine the evolutionary potential of our world, or at least to dramatically reorient it. Big picture, authorities as diverse as Stephen Hawking and Monty Python have observed that humans are really just a minor organism on a planet orbiting just one of billions of stars on the outer spiral arm of a galaxy we call the Milky Way, which Hubble tells us is only one of billions of galaxies in the universe. Furthermore, all life as we know it is embedded in a precious, thin film or membrane called the ecosphere, which envelops our Earth, something like the skin of an apple. This means that life is not so much a spark or property that is inherent in individual species. Rather, life is inherent in what surrounds and makes species possible. Therefore, think of your life as a gift from all that surrounds you, your environs or environment. No surround, no you. No surround, no life on Earth. So, astronomically speaking, we humans may be small stuff, but our impact on our life-giving home planet has been inordinate, both suicidal and worse, biocidal. We're killing ourselves, and we're taking much else with us. The Earth and humanity deserve better. By incorporating the two species conservation strategies I have suggested tonight, I believe we can do better. First, give equal emphasis to sustainable use and protection. And second, go deeper to conserve not just species, but that which makes species possible, including ourselves. I'll close with this well-known nugget from Monty Python's The Meaning of Life. Remember, when you're feeling very small and insecure, how amazingly unlikely is your birth. And pray that there's intelligent life somewhere up in space, because there's bugger all down here on Earth. Thank you for proving Monty Python wrong. Thank you all for representing intelligent life on Earth. Thank you.
Questions? Time for 2 questions. Anybody? Gonna let him get away with that? Somebody, aha, somebody's rising to the bait.
Comment with respect to you're saying about crocodiles. If we get to that state, that's a very idealistic state, because although the world be here, human beings at that point won't be able to breathe or live on the earth. So having been studied this for quite a long time, I understand where you're coming from, but we need to stop getting to that, but stop before we get to that point, because we won't be here. And with respect to, you said, if I may ask you how you achieve this, how do we stop the explosion with population of human beings? That, I think, is a fundamental problem. Thank you.
Well, I take your comment, and I realize it's not a question. One of the reasons I said undermine the evolutionary potential of our planet, or at least reorient it, is that I think the Gaia hypothesis would suggest that it's really impossible to undermine the evolutionary potential of our planet. It will retain its evolutionary potential. The question is, what are the life forms that are going to evolve? And that may or may not include us. And I think that's relevant to your point.
Another question?
Monty, thank you for a very deep presentation. I'm wondering how you feel our nation, Canada, is doing in terms of adopting the first strategy that you've described.
The protection strategy?
Dealing with our development of our environment in a sustainable fashion.
Did you mean, when you say the first strategy, do you mean the protection strategy?
Exactly.
Well,
protection versus your
sustainable
second
use.
option.
Well, that was actually not an easy talk for me to give, because I've spent 42 years advocating protection and protected areas, and excluding as much as possible, certainly, industrial use. And by all measurements, I mean, Canada's made international commitments to protecting certain percentages of our ocean, of our lands, and we're miles behind on all of them, which is a shame for one of the remaining big green patches on the planet. Most people who don't live here still do think of Canada as a wild and beautiful country. However, I could give 2 talks tonight. I could rhyme off all the gains we've made. In the 1990s, we launched something called the Endangered Spaces Campaign. We doubled the amount of protected area in Canada. We set aside over 1,000 new parks and protected area for unprecedented progress on the protection agenda. You know, if I actually could have brought slides and pictures and just knocked your eyes out with the progress we've made. I could talk about what I think, I'm a forester by training, what I think have been some very deep changes in the forest industry in this country where they are trying seriously to move towards sustainable use of that resource and accepting that there needs to be sizable protected areas for proper biodiversity conservation. So it almost, you know, on the other hand, I could give you the David Suzuki talk on how we're going down the dumper and how much we've lost, and that was really the drift of the numbers I gave you globally from the Living Planet report. So your head would swivel and you'd say, well, which is it? You know, are we making gains or are we losing? Well, the answer is yes. We are making gains. You insert certain areas. We are making gains. And yes, we are losing. The question is, which is outdistancing what? And it isn't that we aren't making gains, we are, but our losses are outdistancing our gains. So it's the rate of change, it's not just change, but the rate of change and that imbalance between losses and gains that I think we really need to be concerned about. Thank you for your question.
Thank you. Our next speaker is Stuart Pym, who will continue to explore the themes. The floor is yours.
I have two missions tonight. One is to save the planet, and the other is to sell you indulgences for the miserable sins that you are committing in terms of the carbon that you put out as global emissions. I propose to do both of those things in 20 minutes. I want to look at three questions. What do we know about species extinction? How can we use science to prevent extinctions? And then, oriented to the barcoding group in the audience, I want to consider very briefly what the limitations of our science are. Studying extinction is like studying death rates. And we can measure how fast species are going extinct by looking at how many obituaries that we see, if not exactly in the newspaper, but in the accounts of species when they disappeared, how many disappeared. And we express those death rates as extinctions per million species per year. It's rather like a death rate for people. If I wanted to know whether it was safe to visit Guelph, you know, I would expect the human death rate between, 10 and 20 people per 1,000 per year. If I came across crosses row on row with poppies growing in them, as both of my grandfathers did on the Somme 100 years ago, I would know that the death rate was too high. So if we look at the death rates of species, they're on this order, 100 to 500 extinctions per million species each year. And the question, of course, is that high? And the answer is, well, yes, it's very high indeed. We can use a variety of different kinds of sources, a fossil record, molecular phylogenies, a lot of other pieces of evidence to work out how fast species ought to be going extinct. And the answer is a death rate of about an extinction per 10 million species per year. That means that we are driving species to extinction 1,000 times faster than they would be going extinct without our efforts. I really dislike the term the sixth extinction. It has an air of finality to it. It suggests it's all over, that you should leave this room depressed and throw yourself off a tall building. And I don't want you to do that. I want you to come away feeling empowered, feeling that there are things that we can do to prevent that sixth extinction from taking place. So the first take-home message is we know how fast species are going extinct. We know how fast they should be going extinct. It's too high, but as we will see, there's a lot of things we can do to prevent those extinctions. So how can we use our science to do something about this? How can we prevent this sixth extinction from unfolding?
The number of species of birds and mammals and amphibians we can now map out with some considerable detail. These are birds, and they show that the greatest numbers of species are in the tropical moist forests of the world, a pattern that covers all the continents. The interesting and unusual aspect, however, is when we begin to look at species in terms of their geographical range sizes, look at the species that are smaller than the median range. Those distributions are profoundly different. They are concentrated in special places, what my colleague Norma Myers calls hotspots. And the Northern Andes is a hotspot with extraordinary numbers of small range species. Now, small range species are the ones that are at the greatest risk of extinction. So conservation is very much a matter of looking where those species are concentrated.
If we look at where species are on the verge of extinction in the Americas, we see that they're concentrated in two places. They're concentrated in the Northern Andes, and they're concentrated in the coastal forests of Brazil. And the examples that I'm going to give you tonight come from those two places. These are birds. We can do this for birds, we can do that for mammals, we can do this for amphibians, and the patterns are broadly the same.
The reason that there are so many threatened species in the Western Andes is obvious when you look at the satellite imagery. Very little forest remains. The habitats and the valleys have been cleared for agriculture. But it's worse than that, because what remains are habitats that are fragmented. And this area, the south of Jardine, is a particularly worrying example of that. You can see that the remaining forest has been broken and fragmented, and a large area, about 100 square kilometers, is in danger of being pinched off from the rest of the forest in the Western Andes by deforestation creeping up valleys to the north and the south of this very tenuous forest corridor. That is something we need to change.
We know a good deal about what happens to species in habitat fragments, in habitat splinters. And we know that from the most extraordinary ecological experiment ever done. I think it's now in its 40th year. It's the work of Tom Lovejoy, who is on his way back as we speak from this very location, and I believe will be talking to you tomorrow night. We know what happens to species in small fragments. They go extinct, and small fragments lose species very rapidly indeed. The smaller the fragment, the more species that a fragment will lose, and the faster it will lose them. So when we look at the nature of landscapes, places like the Western Andes, coastal forests of Brazil, Madagascar, other parts of the world, the extinctions taking place there are because the individual populations in fragments are not viable. We can do something about that. One of the things we can do is simply go to those areas, engage the NGOs in the countries, Colombia in this case, and we can help those international NGOs buy up and protect their own land. I'm very sensitive to the fact that we gringos don't need to be going into Colombia and telling the Colombians what to do, but there is no reason why we shouldn't help Colombian NGOs buy critical areas of land and reforest those lands and reconnect those areas. Oh, that animal is the olinguito. It is a gorgeous, cuddly little animal, and I'm sure many of you have seen this photograph. It went viral last year after the olinguito was discovered. That photograph of a baby olinguito comes from that area outlined in green on the slide up there. So what we have done, my organization, Saving Species, has raised money to help a Colombian NGO, Fundación Colibrí, the Hummingbird Conservancy, buy up these critical areas of land and reforest them. Of course, where do we pick these fragments? Next bit of science has to be for us to decide what the priorities are. Now, the earlier movies that I showed you take a strategic view of saying the Northern Andes, coastal Brazil, Madagascar, the Philippines, places like that are international priorities. But how do we go from that to scaling down to the practical side of where exactly do we work? We can do that, and we can do that by combining high-tech modeling of where species occur, satellite images of where the remaining habitat is, digital elevation models of elevations and knowledge of where species occur. Putting that together, we can create very, very detailed maps of where we think the species should survive. This is the study done for the Western Andes. And this map, which I think is my favorite, is one of an area very close to the city of Rio de Janeiro in Brazil. That big embayment is Guanabara Bay. The city of Rio de Janeiro is off to the left. Now, what this map shows in color are areas that have forest, color-coded by the number of threatened species that occur there. Red has more threatened species, blue has fewer, and draped over a three-dimensional image of the area. When we did that, we noticed that the greatest number of species in the map, indeed the greatest number of species in all of the Americas, was a group of forest fragments at the right-hand side of that image, at the east of that image. And we thought we could do something about that too. We can prevent the species in those fragments from going extinct by buying up the intervening land, the cattle pastures, and reforesting it. So that was our first project. What should we expect? Well, I expected it might take a long time for that reforestation to take place. We raise the money, we give it to our Brazilian colleagues, the Asociação Mico Leão Dourado, the Golden Lion Tamarin Association. They then gave the money to the Brazilian government, and they started planting trees. I thought planting trees might take a long time. In fact, within about a decade, the forest has come back enough to allow the golden lion tamarins and all the other species to move backwards and forwards out of that forest fragment prison in which they once could not escape. So that's what it looked like before we started our project. This is a cattle pasture that divided a fragment of forest to the right that was an island isolated where the species could not maintain a viable population from the forest to the left. And as you can see, within a relatively short period of time, about 7 years, the forest has begun to fill in, and the species can move backwards and forwards. That's what it looked like in the beginning. That's what it looks like now. We can connect, protect, and restore ecosystems. And by maintaining, restoring that integrity, we can dramatically reduce species extinction rates. What's the problem with this science? Well, the problem with this science, the problem with setting the conservation priorities that I've talked about, is it's all based on birds, mammals, amphibians, a little bit on plants, and we know that that's a very, very small part of creation. We know that there's a lot more species out there, and what worries me is, while I think there's a general argument to be made that the coastal forests of Brazil, the Western Andes, Madagascar, these other places are the international priorities, do we know that they are the right priorities for butterflies, for fungi, for all these species that we don't know very well because they are hard to work on taxonomically? So, to you barcoders, we do not need to know all the species, but we do need to have good geographical surveys of a wide variety of taxa. At the moment, we're setting our priorities basically on the basis of birds, because people love birds. They express more passion for birds than they do about nematodes. But we do need to be sure that the decisions we're making are representative of all biodiversity. These surveys need to be complete. We need to know where the rare species are. We know that the species that we discovered first tend to be wide-ranging and common, but those are not the species at greatest risk of extinction. And we do need to be geographically comprehensive. We need to know where the vulnerable species are. We need to know where not just the vulnerable birds and mammals and amphibians are, but the vulnerable butterflies, you know, maybe even the vulnerable nematodes. How do we make this happen? Here is where I give you a chance to absolve your sin. You are emitting carbon to the tune of 7 tons of carbon per year. You should feel ashamed, my children. You should feel that you are going to burn in hell because of this. Just for you, this evening, I have a special. For 70 Canadian dollars, currently at a discounted rate, you can absolve your carbon sin for a whole year. So I hope you will join with these usual suspects: Ed Wilson, Tom Lovejoy, you'll see him tomorrow, me, Peter Raven, Pat Wright, Trevor Price, all of whom constitute saving species. We believe that we can connect, protect, and restore the planet, that we can reduce the extinction rate. We can do that by selecting with very careful study the places where species are going extinct and prevent those extinctions from happening. Thank you so much.
Fantastic.
Go ahead.
We have time for 2 questions. Any questions? Everybody eager for a break?
We take all major credit cards.
All right, we're going to break for 20 minutes now to give you a chance to stretch your legs. Please come back in for 20 after, okay? Thank you. And thank you very much. That was fantastic. Even though a few people are still outside, our next speaker is taking us back to birds. Bridget Stutchbury, who's right here in the wings, ready to start. Bridget, floor is yours.
Thank you. If you want to bring people in from the lobby, you just have to say, we're having a bird talk. They'll come right in. Birds are popular, and I think I love birds as much as Lawrence probably loves bees. I'm going to try to be a bit more diverse in my talk, at least at the beginning. My job that I was given was to give a bit of a Canadian perspective on the status of land animals in Canada. And you can see from my title that, yeah, I am a bit bird biased. I'm also known as the bird detective. But there's a good reason to focus on birds, and that is that birds represent a huge biodiversity on the planet. There's 10,000 species of birds, and in Canada, about 450 species of birds, which is quite impressive. Unfortunately, in Canada, when we look at what vertebrates, at least land vertebrates, are listed as species at risk in Canada, officially listed, unfortunately, we have kind of a bleak scenario where 1 out of every 5 are listed as species at risk in Canada. And this amounts to about 150 species. So out of the 700 or so species that we have in Canada, about 150 are already considered at risk of extinction. About half of these are birds. And part of the reason for this is because birds are so numerous in Canada, so speciosh, as I said, about 450 species of birds in Canada alone. But of the species that are listed, about 1/3 of them are reptiles and amphibians. And so to give them their fair due, I just want to spend a moment talking about these reptiles, amphibians, and why they are even more at risk in Canada than the birds that I love so much. So we have about 40 species of amphibians and about 50 of reptiles in Canada, which is good. But sadly, 42% of all the amphibians and 77% of all of our reptiles are species at risk. And so this is kind of a stunning level of risk for these important groups of animals. And you might ask, why are the reptiles and the amphibians so at risk compared to the other groups? For birds and mammals, it's about 15%, to put it in perspective. Well, for reptiles and amphibians, one of the issues that they have that makes them so special is that many of these species are at the northern limit of their ranges in Canada. So I've given a few examples here to illustrate that, where they just barely get into Canada. And unfortunately for them, Canadians prefer, generally speaking, to live in the south rather than the north. So our population is concentrated in southern Canada, and that's where some of the heaviest anthropogenic impacts occur. So we have this intersection of species that occur only in southernmost regions in Canada and have very restricted geographic ranges, very specialized and restricted habitats available, and this is precisely where most of the people are. One way to illustrate this for reptiles and amphibians is this illustration that comes from Ontario Nature, which shows the road density in southern Ontario, which is amongst the highest in Canada. And of course, many reptiles and amphibians are at risk from direct mortality from roads, being hit by cars, but also from pesticides and contaminations of chemicals that wash off these roads, and the fragmentation of the habitat that comes from having so many roads and so much development in this area. So for reptiles and amphibians, they are our most at-risk vertebrates in Canada. And certainly a habitat loss and fragmentation is their most important risk. And road mortality would come in as a close second for many of these species. But there are some gaps in our knowledge, which I won't get into tonight. We don't really have time to review this. But in terms of how pesticides affect many different reptiles and amphibians, we're just scratching the surface in the research to date in understanding which species are impacted by which chemicals and how serious a threat this is. When it comes to infectious diseases, of course, we have things like the chytrid fungus and ranaviruses, which are really well studied. But some of our reptiles and amphibians are currently at threat by new invasive species that are partly the result of the pet trade. And I'm thinking of salamanders in particular, where there are new threats emerging because these species are traded globally and opening up possibilities for new invasive diseases to enter Canada. And when it comes to climate change, again, we don't know a great deal about whether climate change is actually going to benefit reptiles and amphibians because They're at the northern limit of the range, and we expect warming, so perhaps their range will expand and allow them to occupy different types of habitats or more habitat, or whether we'll have, generally speaking, a negative impact on the species. So again, an important gap in our knowledge for the conservation of these groups. But getting to my favorite subject of birds, as I said, Canada has an impressive biodiversity of birds with over 450 species. Shockingly, about 15% of these are already listed as species at risk. And there are dozens of additional species that have been undergoing serious long-term population declines over the past decades, who will be candidates or already are candidates for listing, but have not been listed yet because of the backlog in getting this important work done. So what I want to talk to you about today is some of the threats that our birds face in Canada, and to particularly concentrate on migratory birds. The bobolink is a classic example of a migratory grassland bird, and the graph that you see on the left shows the decline in bobolink numbers in Canada since the 1960s, when we began systematic annual surveys, the breeding bird survey, to quantify just what the problems were. This is sort of like looking at the canary in the coal mine. If birds are declining, it's an indicator of what's happening to the habitat and environments around them. And you can see that the bobolink is declining by a shocking 3.6% per year. If you were investing your money in the bank and was told this is the return rate you get on your money, you would never put your money there. You'd be out of money after a few decades. The same thing is happening with birds. Another example is the olive-sided flycatcher. If you noticed with the bobolink, their breeding range is also concentrated in southern Canada, much like the reptiles and amphibians. But with birds, we have kind of a different picture because so many of our birds are boreal forest birds that have extensive breeding ranges across many of the vast northern regions of Canada, where much of the habitat is still intact. So it's a bit of a puzzle why a bird like the olive-sided flycatcher, which has such an enormous breeding range, is undergoing such steep declines. And yes, habitat loss and fragmentation in the boreal forest no doubt is driving some of these declines, and yet the species still has extensive intact habitat, which calls the question, why would it be declining so severely? And part of the reason is because they're migratory. So what makes Canada's birds so special and different from some of our other vertebrate groups is because the vast majority are migratory. Because they're mobile, because they can fly long distances, they leave Canada in the winter and occupy tropical regions during our cold Canadian winters. And this makes it so difficult to identify what's driving the declines of individual species, and more broadly, what's causing the decline of Canadian biodiversity of birds overall, which are the biggest threats. We need to be able to look at different parts of the life cycle to understand the declines of individual species and groups of species. So if we imagine a bird like the Wilson's warbler, it very well may be breeding in the boreal forest, hopefully an intact forest, but it undergoes, each individual bird undergoes a massive long-distance migration, which would take it often down the east coast of the US, where there's many urban cities it has to dodge along the way. Habitat loss at stopover sites along the US Gulf Coast as it fattens up to get ready to fly across the Gulf of Mexico. And when it finally arrives in Central America a few weeks later, the bird has to find suitable habitat there to survive the 5 or 6 month winter season as it lives in Central America. Come spring, of course, it has to come all the way back to Canada before it can even breed. And so this marathon journey, you can imagine all the threats that lie along the way: breeding, migration, and winter. We talked about habitat fragmentation in some of the earlier talks, and certainly there have been extensive studies of how habitat fragmentation affects breeding songbirds. And really these small patches of forest that are left in our, for instance, our southern Ontario landscapes, like we have around here in Guelph, are kind of ecological traps, if you want to call them that. Birds readily occupy these fragments and breed in them, and yet their reproductive success there is very poor. And there's been numerous studies showing just why this is the case. In these small fragments, birds called cowbirds are able to come into these fragments and lay their eggs in other species' nests. And so in the picture here, you can see these are the cowbird eggs, the larger ones. three smaller white ones are the chipping sparrow eggs. And you can guess in this picture which one is the cowbird and which one are the baby chipping sparrows. Over here we have nest predation. Nest predation rates are far higher in small fragments than they are in large continuous forests. And so we know that forest fragmentation is a huge issue, not just in the tropics for resident birds, as we heard earlier, but also for migratory birds when it comes to breeding here. Cats are an enormous problem. Cats are invasive species. They're not a natural part of our ecosystem. And certainly here in Canada, studies have been done to try to quantify just how much damage are cats doing to our ecosystems, to small mammals, to birds. And these broad surveys, some of the techniques they use is to ask the cats to go pro and wear little cameras on their collars. And as the cat goes out feeding at night or in the daytime, they're able to record all of its activities. And you can see here one example, and if you really want to watch it on YouTube, they have this video of this poor Phoebe getting attacked and played with by a cat and eventually dying. It's estimated that 200 to 300 million birds per year are killed in Canada by our cats. This is a huge toll that it's taking on many, many different species. One of the big mysteries that we've had to solve when it comes to migratory songbirds is trying to understand what threats they face on a migration and on their wintering grounds. And until 5 or 10 years ago, this was largely a black box because there was no way to track individual birds on this epic journey that they were taking. But now we have these little tracking devices called geolocators, and you can see me holding one in the picture up there. And we can put these on the birds as a little backpack that they wear on their back, and they go off on migration. They come back again the next spring to their breeding site where we can catch them, take off the backpack, download the data, and create these maps of the timing of the movements that this bird made throughout its whole journey and identify the core wintering sites. So we've heard already that tropical deforestation is a huge issue, and certainly for our Canadian birds who migrate to Central and South America, they face extensive deforestation when they arrive. Now, with these tracking devices, we can identify what those core wintering areas are for particular species. So in this example, we put tracking devices on wood thrushes in Waterloo, not too far from here, and east of here in Kingston, to try to find out where the Canadian wood thrushes spend the winter. And in this case, the answer was in eastern Honduras and Nicaragua. And if you look at the state of the forests in Nicaragua, just as a for instance, massive deforestation has been taking place, especially along this eastern part. So when we talk about how to conserve individual species, especially something like a Canadian wood thrush, the answer lies far away in Nicaragua. And it's only by connecting the breeding sites with the wintering sites that we can really start to solve the puzzle of why Canadian wood thrushes are declining so dramatically. We can also look at the migration routes of these birds as they fly back and forth, because it's thought that most mortality that happens on this entire journey happens during migration itself, that's the riskiest part of their trip. Once they're at their breeding sites, the adults are fairly safe. Once they're at their wintering sites, they're relatively safe. It's going back and forth, which is so dangerous. So we map out what these routes are. We've discovered in the wood thrush what we call a migratory bottleneck, that most of the wood thrushes that breed in Canada in the northern part of their range migrate through the New Orleans area, the Mississippi River Delta, right here. So we know that that area is an important site to target for conservation, because overall, across all the wood thrushes that we've tracked, it's estimated that about 65% of all wood thrushes in North America funnel through that narrow area in spring. So targeting that area for conservation and having decent forest patches for those birds to stop over and refuel is an important way to protect this species as well. On the right, you can see a typical migration loop for Canadian wood thrushes. In the fall, they come down the eastern coast of the US, and in the spring, they cross the Gulf of Mexico and up through the Mississippi River Valley. The background is light pollution at night. These birds fly at night. They migrate at night. And in some ways, it's amazing when you look at this picture that they can even survive the migration at all, given the heavy human footprint. And I would just note down here that this little dark area down here is the core wintering site of wood thrushes. which is still intact, as I showed you earlier, is being rapidly deforested. Lights have a different kind of problem because birds are attracted to the city lights during migration. And in the daytime, they're faced in living within an urban environment or somebody's backyard. And as they start foraging, they fly into windows. And again, we've been trying to tally up just what is the total damage from this. It's not nearly as bad as cat mortality, but it's estimated that some 25 million songbirds are killed in Canada each year from these window collisions. Many of this happens as birds fly through in massive numbers in fall and spring migration. But the good news is that some of these small forest patches that we have, which are completely useless for breeding, in fact, they're ecological traps. Birds breed there, attempt to breed there, but hardly produce any offspring at all. Those same forest fragments, which are horrible for breeding, are excellent stopover sites. So here's an example of where I work at York University. There's a little woodlot across the road from the biology building. And during fall migration, we go out with my students, ornithology students, and we catch all these amazing boreal forest birds that would never be caught dead trying to breed there. It's way below their breeding range. The forest patches are way too small. But during migration, that tiny woodlot is full of birds like brown creepers, Swainson's thrushes, black-throated blue warblers. Dozens of different boreal forest birds use those kinds of woodlots as stepping stones on their migration. So if we can conserve even these small patches for migratory birds and create a network to allow them to migrate safely, that can be a huge benefit. So when we look at overall, what are the threats to Canada's migratory birds? Certainly, number one, habitat loss and fragmentation. So protecting large extensive habitats for those species during breeding, and then smaller habitat patches and more urban, suburban areas for migration stopover are really important conservation tools. And invasive species like cats are enormously important. And when we step back from the Canadian perspective and look at the global view, these same two factors are the most important threats to birds worldwide: habitat loss and fragmentation, and invasive species. This map is the same one that Stuart Pym was showing you earlier, basically from his paper. And it shows the global diversity of birds around the world. And I just want to mention briefly, because I haven't really talked about DNA barcoding at all, when it comes to our North American bird species, we have identified most of the 450 species in Canada very easily by morphological cues, singing behavior, genetics, etc. And so the role for DNA barcoding and species identification within Canada for birds is not a very big role. But that's not to say, of course, that it's not important when you step back and take a broader view. There are many examples where people are using DNA barcoding to do what we've heard of already in this series of talks, identifying cryptic species. So in the Philippines, for instance, you have a species like this one here that appears to be widespread amongst different islands. But when you do the DNA barcoding, you discover that it's actually 3 different species, likely, rather than one widespread one. So this sort of work is happening in areas around the world where the birds are less well studied or the areas are less accessible to help us understand what is the true diversity. And in this study site, it's been estimated in the Philippines with these birds that the actual biodiversity in this area of birds is 50% higher than what we already know based on the DNA barcoding. So it means that this area is even more of a hotspot than we realized before. And also there's been a lot of diet studies, applications for DNA barcoding, looking at what birds eat by examining their feces. And the one that I found most interesting, which I'll close with, is an example where they're actually looking at the seeds, collecting seeds off the forest floor and extracting DNA off the coating of the seed to find out which bird actually ingested the seed and pooped it out the other end. So it's kind of putting the diet studies on its head, so to speak. Thank you very much.
Fantastic. We have time for 2 questions.
Canada shares a unique responsibility for the tundra and the Arctic ecosystem. I'm wondering what your opinion is on how that's going.
Well, certainly when it comes to climate change, those areas are most vulnerable. And I think some of, even for our migratory birds that breed up there, there's good reason to think that that's going to be a major driver of continuing declines of our Arctic species, because those are the areas where the climate is expected to be changing the most. And for most of our Arctic birds, they're probably least studied compared with the birds in the deciduous forests of southern Ontario. So again, we have a gap in our knowledge, plus a likely very large threat for those species.
Any other questions up at the front?
Hi, I'd just like to ask you if you could speak to bird mortality in wind turbines. I've spent a lot of time at Pelee Island, and on my way through Jack Miner area, there's a little sweet spot on your map with road density that's fairly clear, but that area is really high density of wind turbines now. I'm just curious, have you got any research on or data? Or?
Yeah, I mentioned in my talk that people have, there were a series of papers published recently where they took the best science, best available information and tried to quantify total mortality from different human-caused events. Cats were number one with 200 or 300 million a year killed in Canada. Not the cats, unfortunately, I should say, the birds. And then when it comes to window collisions, as horrible as they are, we've all had birds fly under our windows and we feel horrible. Again, that adds up to something like 20 or 30 million a year, which is a lot. You do that same exercise with wind turbines. How many birds are being killed per turbine? How many turbines are there in Canada? The total mortality per year is in the neighborhood of 25,000 birds, which is, it's a lot of birds. And it doesn't mean that we can't mitigate that and make it 5,000 or 0. But compared to other things like window collisions and cats, it pales in comparison. And it's not to say that there aren't other reasons not to have wind turbines in particular places, but when it comes to migratory birds, they don't seem to be a huge threat from that.
Thank you very much.
Thank you.
Our next speaker is Paul Snellgrove, who's going to talk about oceans of biodiversity.
Well, thank you, Brock, and thank you all for hanging in. I know it's been a long day for many of you. I have some really tough acts to follow tonight, but in fact, they set up some of the points that I want to make, so that actually works out quite well, speaking towards the end. I have the honor and the privilege tonight of representing the oceans, the 70% of the planet that most of us don't think about much on a day-to-day basis, but of course play a critical role in a whole variety of aspects of the health of this planet. So I'm going to start off with some good news, bad news, and then I'm going to spend most of my time on more positive notes because we want to end positively. So my first bit of good news is that the number of documented extinctions globally that I know of in the ocean is 11 species. So we can pat ourselves on the back and think that sounds like it's really great, but there are two words that I use there that you should consider very carefully. One is documented and two is global. And so by documented, I mean species that we know exist and we know we've driven to extinction, and so there's an issue with that, which I'll demonstrate in the next little while. And the second point is global. We've done lots and lots of local extirpations around the world, and we've certainly decreased biological diversity in the oceans in terms of genetic diversity. So let's not get too complacent about what's happening in the oceans. Now I go to the bad news. The bad news is that the rate of deterioration of the oceans and the effect that we as humans are having has accelerated and continues to accelerate over time. And so the culprit in the past was primarily fisheries, which continues to play a key role, a whole variety of aspects of fishing, overfishing, habitat destruction, food web modification, a whole variety of issues continue to modify and change oceans in a negative sense. But more recently, we've superimposed climate change upon that fishing impact in terms of major worries about life in the oceans. And so the big thing, of course, with climate change is that every drop of water is affected by what happens in terms of climate change. We have changed the pH measurably of every drop of ocean in the world, which is staggering to think about. But let's get on a more positive note. Tonight we want to talk about biodiversity. How do we assess it? What do we know about it? And how can we conserve it? I could spend the rest of my time focusing on the negative, but On a positive note, I was part of a program from 2006 myself through 2010. It was a 10-year program called the International Census of Marine Life. In fact, there are several people in the audience here who were also part of that program. And one of the things we discovered through that program is new species pretty much everywhere we look, from the coastline to the deep ocean, from the Arctic to the tropics, and from microbes all the way up to fishes and everything in between. And in fact, it turns out that the constraint on new species description is the taxonomists, the specialists who describe these species. And so the number of new species described for the oceans is now 4 to 5 species per day, which is pretty good. I'm going to come back to that number a little bit later. So that raises the question, what don't we know? Even after a 10-year program like the Census of Marine Life, we know that there's a lot of undocumented diversity in the oceans. Now, this is a project out of Dalhousie University. Camilo Mora, as part of his postdoc with Boris Worm, did this work trying to extrapolate how many more fishes are there out there to discover. Now, fishes are an interesting group to work with because, in fact, they're pretty well known. In fact, other than marine mammals, they're the best known group of organisms in the ocean. And so by fitting these different models, and some of them are familiar to you, I expect, if you've done any chemistry, because we're starting to see a decline in terms of the rate of new discovery of new species, these discovery curves can allow us to project how many more species we'll find before that discovery curve levels off and we stop finding new species. So in fact, through that exercise, they estimate that there is something like 16,500 or so known species of fish and another 1,000 to 4,000 more species to go. So a group we know really well still has some pretty significant discovery ahead of it. Now fishes, as I said, we know pretty well. Now, Stuart Pym talked about nematodes and pointed out that most people don't care about nematodes. But this is a discovery curve for nematodes in the deep ocean. And the point here is that if you look at whether this is going to level off, you could say, well, maybe it's about to level off, although there's no signs of it. You could say it'll level off in a little while, or you might even suggest it'll go through the roof of this building before it levels off. In fact, if you talk to deep sea nematode specialists, or nematode specialists in general, they predict they have 100,000 more species to go. Now, as Stuart said, nobody really cares about these nematodes. They're little threadworms that most people don't even know exist. And a very well-known marine taxonomist by the name of Philippe Boucher, who does mollusks, which people do care a little bit more about, made the comment that people will be more excited about that last species of fish than hearing that we've discovered 100,000 more species of nematodes. And it's just the way human nature is. Now, one other point is statisticians, actually Andy Solo and Wolcott Smith, made a really interesting prediction, namely that there are still 16 animals greater than 2 meters in size left to discover. So it's not just little stuff that's left to discover. There's big stuff out there as well. So that's really interesting to think about conceptually. Now, there we go. Okay. Now, one of the other interesting things about the ocean is that it's opaque. We tend to think of water as being clear, but in fact it is not. And we're still discovering new habitats in the ocean. Now, some of these will be quite familiar to you. For example, hydrothermal vents. Some of you may even know about cold seeps. But there are these new environments such as brine pools in the deep ocean, pockmarks on the seafloor. And if we look at the discovery curves for new habitats, in fact, they also are not starting to level off. So there's lots and lots of new habitats out there. And of course, with new habitats, we always find lots of new species. So again, really good potential for species discovery. And so this red line here is just one I fitted by eye. But the point here is that it's just not showing signs of leveling off. Keep in mind that hydrothermal vents, for example, really revolutionized the way we thought about life on Earth because they're completely different ecosystems in terms of how they're run. They're dependent not on sunlight, but on chemosynthetic energy. So you might make the argument, well, is it just stamp collecting? So lots and lots of unknown species, sure, that's great. Does it really matter? Well, in fact, it does. And this point was made briefly by one of the previous speakers, namely that we know biodiversity are important drivers of different ecosystem functions that are really important, including nutrient cycling, habitat for other species, climate regulation, a whole variety of issues. These in turn, in fact, drive a bunch of ecosystem services that we care a lot about, like fisheries that provide employment and food and so on, medicines, cultural value, and so on. Now, the interesting thing about this is that there's feedbacks. And so if we look at the benefits to humans, for example, we derive a lot from it. And unfortunately, what that does for human society is allows us to increase our numbers dramatically and continue to grow in population size. And so a point that we heard earlier, we simply have to do something about the rate of population growth because the oceans just can't support it anymore. Okay. So we have lots and lots of diversity. We have lots we don't know about. What are we going to do about it? Well, it turns out that, in fact, we have better tools now to try and meet these challenges. Now, I could spend a little bit of time on each of these, but I just want to focus on two of them, given the topic of tonight's general discussion. First of all, we have better sampling platforms than we did in the past. And second of all, we have genetic tools that we simply didn't have even 10 years ago. And so that capacity can really fundamentally change the way that we look at biodiversity in the oceans as well as in other ecosystems. And so by better platforms, I'm talking about the fact that historically the way we sampled the oceans was to drop different types of sampling gear over the side of a ship. We'd pull something up, we'd sample blindly and figure out what it was. But nowadays we have a whole variety of tools such as remotely operated vehicles, submersibles. This is digital imagery. This is interesting. It's actually a coral off the east coast of Canada, a deep water coral. You can see that these redfish, again, an important commercial fishery, seem to be having a nap underneath this coral. There's a variety of species associated with it. This is actually a video grab from video. And look at the quality of the image. And so the technology is just getting better and better in terms of how we can sample the ocean. A second example that I want to give to you is the Neptune project here in Canada. Actually, there's a twin project, Neptune and Venus, which is a shallow water version. And the idea here is that we send a fiber optic cable onto the deep sea floor or the coastal sea floor and put instruments there. And the reason that's important is because we have power to the instruments and we get data back to shore. That seems like a trivial problem, but for ocean sciences, that has been a huge issue for a very long time. And so now we're actually getting ahead on that particular question. Now, in the context of biological diversity, there's a really interesting twist that I wanted to mention. This is something called the Environmental Sample Processor, developed by a friend of mine from graduate school, Chris Sholin, at Monterey Bay Research Institute. And the idea here is it's effectively a molecular lab in a can. And so what it does is it sucks in a water sample, and it looks to see whether there are toxic dinoflagellates present. And it does through essentially using barcode type technology. And so the issue here is that some of these particular toxic dinoflagellates, some of the dinoflagellates are completely harmless. The same species, a different strain of it, can kill you. And so we need to know if we have the toxic strain or the non-toxic strain in our waters, so we know whether we can eat the seafood that these animals end up contaminating. And so here we have an instrument that allows us to do this in real time. It can send the data back to shore and say, stop eating shellfish, you've got toxic blooms going on out there. So I think this is really just the tip of the iceberg in terms of how we can apply this sort of technology to evaluating what's going on in the oceans in terms of change and in terms of threats that are actually an issue for us. So we can use this sort of technology to detect species we know about. We can also use it to detect genes that we're concerned about may be active, such as these toxic algae. Now, believe it or not, we don't completely have, we're getting very close to a list of the species in the ocean, the known species in the ocean. And so, in fact, there's been a project underway called the World Register of Marine Species, which has been putting together, if you like it, a phone book of life in the ocean. Now, for those of you under the age of 25, a phone book is a quaint printed document that we used to use to look up names and phone numbers of people, which of course you do with Google today on your cell phones. But in fact, we don't even have that as a paper document for the ocean, let alone a Google version. So WORMS is actually solving that problem, and they've gone through trying to create a coherent and accurate list of known species in the ocean, and their list right now has about 231 species on it. Now the reason the list is so complicated is that we have misnaming, we have double names, misspelled names, so you publish a paper and you don't have one of the letters right, all of a sudden people think you've got a new species if you don't look at it very carefully. So for example, they found there are 56 different names for a single sponge that have been published in different studies around the world. And so they've amalgamated that and fixed it, and so now this is the source for looking up the names of species in the ocean. But there's an estimate that we may still have about 91% of marine animals yet to describe. There's some debate about this. Some people think it's more, some people think it's less. I think it's probably about the right order of magnitude. So let's say we have 91% still to discover. I told you that we describe 4 to 5 species every day. Well, if you do the math very quickly, we have about 1,000 years of taxonomy ahead of us to describe the animals in the ocean. That doesn't even get into the microbes, which the microbial biologists tell us could number in the billions. And so we clearly have a lot of work to do. And it's a little bit overwhelming just to think about. However, we have new tools to address this challenge. And the Barcode meeting that you're all attending, or many of you are attending, illustrates just such a technology. And so in the past, we had morphological taxonomy, which has its pros and its cons, but it's the way we've gotten to where we are today in terms of knowing what we can and can't eat in the world, for example. But now we have genetic barcoding. And so, again, there are pros and cons to barcoding, but the potential to advance taxonomy very quickly is really quite impressive. And so particularly when we combine these two approaches for molecular taxonomy, once we create that reference database, it can be extremely powerful. Now, we can take this one step further, and one of my colleagues in the census, Nancy Knowlton, has advocated this for coral reef systems. The idea of using mass sequencing, the idea of essentially DNA in a blender. And so you throw samples in, you get sequences, and you figure out what diversity is present, even if you don't know what that diversity corresponds to in a complete sense. So you can get actually numbers for species, perhaps, and eventually we can match it up as we eventually catch up with the morphological taxonomy. So that, again, is a very powerful possibility in terms of accelerating the rates at which we describe life in the oceans. Unfortunately, the clock is ticking. As I've said, our impacts on the ocean are accelerating, and certainly we know there's great ocean change. I would certainly make the point, and I think Nancy will probably talk about this tomorrow during her plenary talk, is that in our lifetimes, for sure, and probably in the lifetimes of your children, coral reefs around the world are going to be fundamentally different than they are today. And we've already seen steep declines. But we're talking about a time horizon there of 50 years, not 1,000 years, for those systems to be fundamentally a lot less diverse than they are today. And so that's a very sobering thought. And so what do we do? Well, we heard from one of the talks earlier today that there are two ways to think about this. One is that we manage the way that we use the oceans, although their example is terrestrial. But the same is true in the oceans, so that we think more carefully about how we fish, how we drag fishing gear across the bottom, we try and reduce our carbon emissions and so on. And I would say certainly in those instances, we can think of good strategies for species that we know about. But then we also have those 91% that we don't know about. How do we deal with that? And it's a huge problem. We can't do this on a one-to-one basis. We can't say, let's do this for cod, let's do this for seals, and so on and so on. We simply just can't. We're going to run out of time and space. And so based on that, I argue for a point, again, we heard earlier, that protected areas have to be part of the solution. And the reason for this is that if we protect habitat, then we protect species in that habitat. And that's really important because it means you can protect things you don't know about. So not just the stuff you do know about, but even what you don't know about. Now, Stuart Pym's talk talked about the challenges in terms of knowing where to place these protected areas to be most effective. In the ocean, we're light years behind what's happened on land. We have even less information on which to base that. We also have a second complication, which is that you can't buy tracts of ocean. And so that's both a positive and negative aspect of the story, that the common ownership of the ocean means that you can't say, all right, let's buy a few acres off the coast of Newfoundland and preserve codfish. And so we have to think of other solutions, and we also have to think about the fact that a large part of the global ocean is in international waters. So it's not even a debate within a country, it's a debate between countries to try and figure out how to resolve this. Still, I think that by creating marine protected areas, we at least buy ourselves some time in terms of unknown diversity. And so as you can see, a lot of marine protected areas, they serve different purposes. Some of them are to protect spawning biomass and so on. But for a biodiversity perspective, large areas of ocean floor and the water above it, I see as a very good strategy to try and buy us some time. And so I want to finish up by putting up some positive images of actually oceans off the east coast of Canada. These are some colleagues at Fisheries and Oceans Canada who took these photographs. Is this what we want the oceans of tomorrow to look like? These sorts of seascapes that are both striking and productive and give us lots of goods and services that make human life much more rich. And so I think we've made a lot of mistakes in the way that we've dealt with the oceans in the past, but I also think there's a chance to turn the tide. And so by these different ideas that I've mentioned and some of the ideas some of the other speakers have spoken of, I think we can do better. So thanks for your time, and I'd be happy to answer any questions.
Time for 2 questions.
I don't know whether any research has been done with respect to the offshore mining and looking for oil and that sort of stuff. The fact that the sea has got different levels and they all work like waveguides. And my concern is, and I don't know this from my aspect, is the blasting and the noise that's being created by this, is it damaging or killing fish because the way the underwater transmission of the sound vibrations and the power of the explosions? I don't know whether anybody's really researching that from that aspect. And a second question I may ask, the fact that the Hadley cell movement and that sort of thing, the intertropical convergence zone is altering around the world, and the El Nino effect and all this sort of thing, are you looking, or how is that affecting underwater things with species and movements and things like that?
Thank you. Those are both really interesting questions. The first one on sound in the ocean, that's actually a really hot topic in the last decade or so as to what effect different sounds are having for ocean life. And so marine mammals, we're starting to get a pretty good handle on that. But for other groups of organisms like fishes and invertebrates, we're just starting to do the research now. But if you look at soundscapes for areas, for example, like off Vancouver Island, the Strait of Gibraltar, it's just stunning the amount of noise in those environments. And so it does raise lots of questions about potential impact. So people are working on that very question right now. Your second one was with respect to changing ocean circulation. There is already evidence that changes in surface production, so the conversion of sunlight into chlorophyll in the surface layers of the ocean, has knock-on effects for all of the water column down to the bottom. And we're already seeing that. And so when we see shifts in surface biota in the oceans, which is already evidence, the rest of the ocean is very likely to follow suit very quickly. And so that's one of the bizarre aspects of climate change, is that if we change surface production in the oceans, it's going to have all sorts of effects throughout the rest of the oceans as well, because that's most of the food for the oceans, what happens on the surface.
Any more?
There's one over there. Oh, Paul's got one.
Well, that was just wonderful. When we heard Bridget speak and we heard Stuart speak of terrestrial environments, patches of habitat, protection of environments, local environments can have a powerful effect in preserving species. You said something that I found really disturbing, and that is that we're altering the pH of every drop of water on the planet.
Mm-hmm.
I mean, this is a system-wide effect. And so many organisms are influenced, their biology is influenced by that shift in pH. How concerned are you about that? How immediate are the effects? And if you are concerned, how do we reverse it or stop it?
So yeah, really interesting problem. So I would say acidification is right now the single hottest topic in marine ecology and the effect that has on ocean life. So far, the evidence is very mixed. Some organisms extremely sensitive, others less sensitive. It raises the question of how quickly organisms can adapt. In some cases, maybe they can. In other cases, they probably can't. So I think it's a very serious issue, certainly for some groups of organisms. The ones with the calcifying activities are particularly vulnerable, but others as well. And this can also go to larval stages. As you know, many species have a larval stage that can have calcareous structures that are also quite vulnerable to acidification. In terms of an answer, that's really a tough one, because that is a global problem. And so, you know, as Stuart mentioned, let's all cut our carbon emissions. Yes, we have to do that, because short of doing that, I don't know how we stop ocean acidification, because it's a freight train, and no matter what we do, it's going to continue to get worse in the short term. The best we can hope for is to slow it down and reverse it in the long term. And when that happens, a lot of change may already have taken place.
Thank you very much. Yes, thank you. David Hick is our next speaker, and he's going to talk about biodiversity in the Arctic.
Well, good evening. The Anthropocene is upon us, and perhaps this is evident nowhere more than in the Arctic. I'd like to talk a little bit about diversity and my interest, which is in understanding the ecological functions of biodiversity in these regions. So there's really three things I'll address. One is sort of the some of the consequences of rapid warming. Give you an example of the types of species interactions that we've started to look at where we've been able to benefit from barcoding and a better understanding of biodiversity in tundra areas. And then finally, I think we should be optimistic that we're really on an accelerating curve for better understanding of biodiversity in Arctic regions. So I think you're all aware we're facing a warming Arctic. In fact, polar regions, the Antarctic and high mountains are all experiencing the same rapid warming. This is very much, there we go, a function of what has been referred to as amplification. Feedback processes operating at both the local scales and through latitudinal heat transport that are leading to much greater rate of warming in higher latitudes. So we're seeing this, and it's pretty much entrained, and we're going to have to figure out how to deal with it. The implications of this are pretty profound at a local, regional, and planetary scale. I think most people are familiar with what's happening with sea ice. This has been well reported in recent years. The same changes are taking place inland with the loss of winter and spring snow cover. Let me just provide a little bit more detail. If you're interested in what's happening with changes in the sea ice conditions, the National Snow and Ice Data Center, NSIDC in the US, daily updates are the extent of sea ice. You can see here in pointer's gone, but where that blue X is, that's where we were a couple days ago in 2015, which is pretty much as the lowest trajectory since 2012 that we've had in winter sea ice. And of course, this has implications for charismatic megafauna like polar bears, and trying to understand where they might be able to persist in the long term as that sea ice environment disappears. As Paul's just described, there's very significant changes taking place in the ocean as well. Not just shift in Arctic species, but species from the subpolar oceans moving into the Arctic Ocean, both small fauna and flora and fishes as well. Not really well documented at all. On land, the spring snow cover anomalies are equally significant. Let's just zoom in to May. These are the results of remote sensing analyses for Eurasia and North America, and we see anomalies of up to or exceeding 2 million square kilometers of snow cover in May. This has significant effects on the phenology of plant growth and the development of plants, migration, and the other ecological processes. And coupled with this are pretty dramatic changes in what's happening in the frozen ground, in the permafrost as it thaws, with all sorts of implications for biota. One of the most observable consequences on land is what's been called the greening of the Arctic. You can see that it's not quite uniform. The blue and green colors here are the areas where we've seen increase in plant biomass over the summer season. The red areas are actually places where the biomass has been decreasing because of fires or drought or perhaps insect outbreaks. It varies from region to region. But in general, we're seeing increased productivity. And much of this is a consequence of a woody vegetation, trees, and in many of these regions, even more rapidly shrubs, salix and betula in particular. This is one of the sites we've been monitoring on Herschel Island in the Beaufort Sea off the coast of the Yukon. You can see in just a relatively short period of time, salix species have come to dominate, and in fact they've grown from 2 or 3 centimeters to over half a meter in just 25 years. So we see this sort of impact on the Arctic ecosystems at a very large scale. And just in case you think from my previous slide, only CBC North is interested in these changes. Last month when we published a paper about shrubification that was picked up around the world. And there's other things happening in the atmosphere as a consequence. Forest fires are leading to an increase in soot and black carbon deposited on glaciers and snowpack, changing the albedo, reinforcing the positive feedback in the climate system. And we've seen in recent years much more attention to the changes in the polar vortex and crazy weather at lower latitudes, and in fact even enhanced warming in the Arctic. So all of these things are going to affect plants and animals and people who live in the north. Now, in the last few years, we've had a very comprehensive assessment of the state of Arctic biodiversity. This was carried out, or led anyway, by the Conservation of Arctic Flora and Fauna Working Group of the Arctic Council, with participation from scientists from many Arctic countries and indeed around the world. And it was received by Arctic Council ministers a couple of years ago, and now the question is really, how do we act on that? How do we address the concerns and the shortcomings that the Arctic biodiversity assessment identified? I won't go through it in detail. There's 20 chapters that deal with parasites and terrestrial invertebrates and even indigenous languages in the Arctic. But there were, and it's worth taking a close look at if you're interested in what the state of Arctic biodiversity is. But there were 9 key findings that addressed issues related to species status, climate change, invasiveness, pollution and contaminants, things like that. And I just want to highlight 4 of those because I think they're relevant to what I want to talk about for the next few minutes. The first is that climate change is far and away the most significant factor influencing the future of biodiversity in the Arctic region. The second point that I'll make is just that there are other disturbances, and how those influence ecosystem services that are important to people who live in the north or that travel and work in the north is something that we really need to get a better handle on. The rate of change is pretty rapid, and the influence of those changes isn't well documented. This thing's dying here. The key point in terms of biodiversity is that we just have really scratched the surface on the distribution and abundance and diversity of species that live in the Arctic. And there's a need to accelerate our understanding of the biodiversity that's there and where it's found and what it means. And that we really require international cooperation to do this, that it's beyond the scope of any single organization or country or group to address these challenges. And in fact, even before the Arctic biodiversity assessment in 2010, there was an effort by Arctic Council to identify indicator species that would be indicative of the changes that were taking place in the north. And even there, it was very clear that we didn't have sufficient information to address or track the changes of most of the diversity, particularly invertebrates and plants. We had good information for harvested species and some of the charismatic fauna. Okay, so one of the things in the last few years, this sort of set us off to think, okay, well, if we have a good database, can we at least start to map the distribution of certain groups? And one of the things we've been working on through a new collective called the Herbivory Network is to at least try to understand the distribution of birds and mammals around the Arctic region by mining databases. This has taken a couple of years to sort of put all of this together. I think it's a good foundation for trying to understand the diversity of species, birds and mammals in particular, and the implications of some of the changes in the Arctic region. We can't do this for invertebrate herbivores yet. We simply don't have enough information about them. But we do know that invertebrates or herbivores are their ranges are shifting. We know that there's an increased frequency of insect outbreaks. Population cycles, or species that have population cycles characteristic of their dynamics are collapsing, and that migratory species are moving around in the landscape in different ways. We've also documented the changes that are occurring in terms of plant production and distribution over the tundra region. And of course, the interaction between the herbivores and the plants is something that is of continued interest. I point it this way, does that work better? There we go. So one of the things we'd like to do is look at the interactions among species. And what I'd like to just tell you very briefly tonight is something that we've been able to look at in more detail because we've had more detailed information about the species identities in the systems where I've been working for the last 25 years. Now, for the longest time, I was interested primarily in cute little furry vertebrate herbivores like this collared pica or pica. This is the species Okeetona calaris that is the red blotch in Yukon and Alaska. These are small lagomorphs, about 150 grams. They don't hibernate. They collect vegetation during the summer and use that as a food cache over the winter. Based on a lot of the evidence that we've been able to provide, in Canada, they've been listed as a species of special concern because they seem to be very sensitive to changes in weather conditions in winter. But more recently, we've become really interested in caterpillars, herbivores in this system that seem to be increasingly abundant and seem to have an increasing effect, variable, but on the vegetation from year to year. And of course, we can collect them when they're flying, but we're really interested in their role as herbivores. One of the species that we've paid some attention to recently is this Arctic moth, Ganophora greenlandica, which is the black dots you see in Greenland and in the Canadian high Arctic. Typically, that's where it's found. And we started to pick up new records in the Ogilvie Mountains and down into the southern Yukon. And when we looked at trying to better identify these guys, we found that indeed they actually came out as a separate subspecies, but related to Greenlandica, which was kind of interesting because we had a fair bit of information about Greenlandica from the high Arctic. How did these guys that were persisting in southeastern Beringia, in sort of this ancient refugia, how were they different and what sort of interactions were they having with other species? Sort of useful to know the relationship among these different species when across their distribution when we're trying to understand their ecological interactions with the rest of the system. So one of the things so here's their basic life cycle. They can live 7 or more years, whereas the picas are only living 2 to 3 years. So the longest lived herbivore in this system that I'll show you in a minute are these little caterpillars, not the larger furry cute mammals. And of course, they lay their egg masses, and the one thing that they don't seem to have to the same degree in the southern part of their distribution as in the north are these parasitoid. There we go. They disappear. So the thing we were interested in is really dying on me here. Can you just advance it? Is that easier? Okay, there we go. So what we were interested in was the interaction between the picas and these moths, and to the extent that they share food resources, what does that mean for this system, and what could it mean in the future as things continue to warm? We've been losing about 11 to 14 days of snow per decade in this particular area. So they share the same patches, the same small patches about 30 meters in diameter. The caterpillars are active early in the season, and then the pikas are foraging much later in the season. And I'll just, if I can, show you a little experiment. You can see a pika down here at the bottom, and there's caterpillars in the lawn. And if we manipulate the system for increasing or reducing herbivory, we could look at the interaction between them. What we found is that if we increase the number of caterpillars experimentally, they eat a lot more of the vegetation, and that's good. That's what we wanted. The following month, we could measure the pika herbivory, and where they were foraging was a bit of a surprise. They like to go to the places where there's more caterpillars. There's a really interesting positive feedback taking place that's mediated through the frass that's produced by the caterpillars, something that we hadn't seen before. And as these guys become more abundant, could become a much more interesting part of this system. So, where does that leave us? Well, with all sorts of opportunities to identify the diversity of these northern ecosystems. Like I say, we've scratched the surface. Eyeball's done a great job in starting to increase our understanding, at least in some places like Churchill and a few other sites, but there's an awful lot more to do. I'm just curious, how many people have barcoded Arctic species in the room? How many would like to now? You know, there's lots to do. This is just a little map that you'll see more of on Friday if you go to this session. But the network of people working on barcoding around the world is phenomenal, and I think there's a lot of opportunities to share the knowledge about Arctic biodiversity. Certainly, Eyeball has started that process, continues that process. There are programs like what Paul described earlier on Arctic Ocean biodiversity. So there's a lot of potential. And in the last year, we've been conducting an international planning process to identify the key research questions for the next decade of our Arctic research. And biodiversity emerges over and over again as one of the priorities. So I think there's a lot of potential and a lot of interest in continuing this work. So finally, just to wrap up, warming and the consequences of warming in the Arctic region are pretty profound. Having a better understanding of biodiversity will help us really uncover more of the ecological interactions that species share in the same environment. And we're well underway with the planning for continuing this type of work. So thank you very much.
Fantastic. Once again, we have time for a couple of questions.
I can't, yeah.
Hi, thank you. Can you explain the loss of terrestrial snow, but the greening of the Arctic? That seems like a dichotomy to me.
Sorry, could you repeat the question?
The loss of terrestrial snow, the declining of it, but the Arctic is greening. So where's the water coming from to green the Arctic?
Right. So much of it is coming from is retained in the ground. There's relatively poor drainage in many areas, and if you increase the length of the growing season, you can have a increased primary production in the season. One of the challenges we have is that might be transient. As permafrost thaws, as drainage patterns for surface water change, it could lead to a situation where you in fact have reduced potential for primary production. But at the moment, the evidence seems to suggest there's an increase in productivity, and part of that is also a shift in the distribution of the woody species further north.
Other questions? All right, thank you very much.
Thank you.
So I need to introduce myself to make observations about one really important link that we haven't talked about so far with respect to biodiversity. And it seems to me that the really important link is somehow getting kids involved and being interested in biodiversity and being involved with it. And the interesting irony, of course, is that more and more kids grow up in cities where they don't necessarily have that many opportunities to interact with nature. And of course, an obvious thing in everybody's garden, for those who have gardens, are insects. You go into your garden and probably find all sorts of insects that perhaps you're happy to see and perhaps you're not happy to see. But of course, usually you don't know what the hell they are anyway. So it's an interesting opportunity to reach out and involve, and it's something that the people here at Guelph have been heavily involved with. There's an astonishing array. The previous insect is a carrot wasp. I'd never heard of them. I found in my goldenrods. And this is, of course, just an ambush bug. And this is the kind of thing that is a very neat way to engage especially young students with the whole idea of them discovering things, as it were, in their own yard. I'm sure most of you realize that if you go out to schools and talk to kids about your organisms, whatever they are, and ideally take your organisms, it's very easy to get a very strong buy-in from the students, and it doesn't seem to matter where they are, and probably doesn't even matter so much how old they are. The opportunity for them to meet a scientist and talk to them and to understand something about organisms is really pretty important. What's really important in all of that is the students actually having a chance to see something and perhaps touch it. You can often wonder about the public health implications of that, but it's still a very powerful thing to do. Now, it's an interesting challenge for people who mainly perhaps work with insects, because some insects lend themselves to be shown to kids and some don't. So I know the Barcode of Life project here has started a school malaise trap. Oh, this is not working. Oh, there we go. School program, and this covers far more than schools. And the idea is that elementary schools, students get a malaise trap, they put it up in the schoolyard. They get to service the trap, they get to empty the bottles to see what animals they've caught. They can send their specimens here to Guelph. They get barcoded, so the student then gets back the name of the insect and a photograph of the insect. And this buy-in has involved over 200 schools, close to 300 schools. And of course, you go from a pile of unsorted insects to a bunch of insects in a collection. I'm told that one of the most exciting things for the kids, the participatory part, is to think that the insects they took out are ending up in a science collection here, and they're important specimens for scientists to study. So it's this kind of outreach that we should all make part of our efforts to promote biodiversity, because if we can get kids to be involved in it, we'll probably have a much easier time in selling the whole idea of biodiversity and protecting it over a much larger area. And obviously something like that that starts early is something that perhaps is contagious. So I could mischievously ask, how many of you got started in the whole game by being involved with it as a kid? And of course, a lot of that's dinosaurs, but I know I have to put up my hand and say, for sure, that was a big thing for me. So we should remember that. And if you get an opportunity, excuse me, to go and talk to the kids at your kid's school or your grandchildren's school, don't miss the opportunity. If you have an opportunity to do that sort of thing at campgrounds and provincial parks, there's a lot of these places are hungry for programs, and they just love to have you come. And I know it takes time, but it seems to me that it's a very important investment in biodiversity. It's always fun to watch kids interacting with animals they haven't seen before, and it seems to me that it's the kind of thing that makes good memories and also draws people in, perhaps not to become biologists, but perhaps to have some appreciation of biodiversity. And I think that's my last slide. So I'd like to finish on that note, pointing out that it's something we haven't talked about overtly, although it has been alluded to several times, and it seems to be critically important. I also understand that there are some buses waiting for us outside. I have a proposal for further questions of speakers that we plan to just meet in the lobby here and allowing you to ask questions of the person you want to talk to. And that would allow for more interaction. If everybody agrees with that, well, I hope you agree with that. We'll just do it. But I'd first like to ask you to join me in thanking the speakers. And we've already thanked the people at the University of Guelph and the Barcode for making the whole event possible, and I guess we can thank them again. So we'll meet you in the lobby. Please come with your questions. It'll take us a couple of minutes to unwire and get there. Thank you very much for coming.